The reason for today's gathering is International Gulf of Alaska Expedition. So this is expedition we just hot of the press, and I'm sure many of you have seen many of us on TV and on the radio and in newspapers everywhere. But this is expedition we just happened and ended up about a week ago. We landed here, we returned to Vancouver on the 18th of March. And we would like, Brian and myself would like to share some thoughts, preliminary results, and maybe initiate some questions and discussion about that expedition. So this is how it's been pitched. So it's International Gulf of Alaska Expedition. It was one of the major contributions to the International Year of the Salmon, and it was funded by lots of people. It was organized and spearheaded by Dick Beamish, DFO Emeritus. He's retired, but he's so passionate about salmon that he dedicated the last of his about maybe a year and a half or 2 years putting this all together. And you can see there is a bunch of people who donated to this expedition. So the total cost was about 1.3 million. And as of today, I just came back from a teleconference with the organizers and everything. We have something like 4,000 in red, which is not bad. But not all the bills came in yet. So I would assume it would be probably about 50,000 in red. But anyway, so it's actually quite good. So it was also organized and in many ways by the North Pacific Anadromous Fish Commission. Which has the International of the Salmon as a project among one of the most important projects which this organization is working on. So we done it on this boat, which is Professor Kaganovsky. Yeah, Professor Kaganovsky, and this is official International Gulf of Alaska Expedition. We don't really call this, but it's part of the contribution to the International of the Salmon. And this is the old photograph there, this is current photograph there. Looks much better than it's actually.
This is the good side.
First of all, good side and good light, and kind of, I took this picture.
Don't look at it from the rear.
Yeah. No, but it's about 63 meters long ship. It's about over 30 years old, about 33 years old. It looks a little bit iffy in places, but everything worked. And we, for example, didn't use a single station or single day to the mechanical problems in this expedition. So, although it's old and very basic, it did work.
I think this is actually his 40th anniversary. It floated in 1978, so it's pretty old vessel.
Yeah,
it was interesting because when we came back, next to us was one of the Canadian Coast Guard vessels, which I think was built in the 1960s. And it was like chalk and cheese, this beautiful Coast Guard. It's been so well maintained, and this one has just been run into the ground. And obviously worked very hard. So,
yeah, I mean, this is a research ship in the Tingo Center, which is a regional fisheries institution in Russia. And the point is that this ship goes from expedition to expedition. It's like sometimes maybe a week or two in between, but otherwise it's almost all the time at sea. And the reason why it looks so well is because it just been out of the docks before the expedition. So it's about 2 months out of the docks. So this is the scientific team, which was with all the gear we need, and the Dick Beamish, who was in spirit all the time at the expedition, and he is organizing. This is our cat, which is about 60 years old, and was from the day one in the ship. And
lives in the oceanography.
Lives in the oceanography of that. It doesn't go much, and very kind of temperamental, but very nice guy. Okay.
Dick Beamish had just watched Life Aquatic, and he thought that we should have matching kit, right?
Yeah, and the one thing which is critical here to say that it was a very international expedition. It was one of the goals, as you will see in a moment, of that expedition, that all the Pacific Rim countries who are working and passionate about salmon would actually have their representatives, and we will all work together, combining our knowledge and also learning from each other. Because there is an idea to have a pan-Pacific expedition in about 2 years' time, where about 4 or 5 ships will be working together and covering at the same time the whole North Pacific. So there was Canada and Russia had the largest representation, and there was 3 people from the United States and 1 person from Korea and from Japan. Okay, so there were 2 major objectives of that voyage, and this is just to say that first of all we wanted to go and see where the salmon is and whether or not we will be able to catch salmon in the North Pacific during the first year winter. This is the continuation of the early marine survival hypothesis. Like you know, most of the mortality is set at early marine stage of the salmon, and the first year in the ocean considered to be part of that. But so far to date, all the research which been kind of directed to this was done on the shelves. We never ever went to into the open ocean. So by saying that, I would like to say that every single piece of information we're going to present today is novel. Nobody ever has done it. So it's the first time, first data set, it's a baseline. And the second, of course, was international team, international collaboration. And it was kind of, in the beginning, a little bit weird because people were talking different languages. And but in the end, we actually had a very, it's a team. Everybody worked like a team, and we actually enjoyed each other's. And this collaboration, I presume, would actually be long-lasting legacy of that expedition.
So before you go on, so just on the second point here, I think any oceanographer will look at this and they'll say, well, every expedition we go on into the ocean is international and collaborative. So this point is particularly for the fisheries, which has, I think it's an important point for salmon, is that the different nations have been managing things very separately and not working together. So this is one of the first instances of having different nations working together on the fishery, on the salmon problem.
Yeah.
Not so much with respect to oceanography. So.
Yeah. The interesting point as well, as I'm continuing that, is that it has been done when 2 ships, for example, from 2 nations would work on the same program. But it has never been done when 5 nations would be in 1 ship working on the same program. That's what just Brian said. That's in short. So this is our survey area, and we kind of north and south separation is in the beginning we've done it, but you will see that it actually does make sense. And this is a particular key and objectives we wanted to achieve. So we wanted to see whether or not we can catch the fish and get eventually the stock distribution in the North Pacific in winter. We wanted to see whether or not we can catch fish and estimate its abundance. We wanted to see whether or not it's healthy and the conditions of the fish, and there were a bunch of hypotheses we wanted to check. Just a few, it's not all of them, because otherwise it would be a long list what we wanted to check.
This is the final survey grid. So this is not what we planned. It's more or less what we planned. Evgeny's going to explain now why we had to modify things.
And it's not actually all the stations, because we did have a few stations here in the beginning, and we did have a few stations on the way down. Yeah. But they were not quantitative and they were not part of the survey. So in total, we in fact did 64 stations, and this is 58 stations in the major grid. And we covered about distance of about more than 9,000 kilometers and area of about 700 square kilometers. So it's a huge grid, and we were able to do this, and surprisingly, right in the beginning, we sat down and we decided how many realistically stations we can do. Original plan was 72. We said, well, maybe 60 stations, it's a maximum we can do. So we've done exactly what we predicted. But why we've done it is because of the weather. We have actually weather not as sunny as this, but as calm as this, about 3 weeks out of 4. So this is the beginning, very beginning of the expedition, when we were just coming out of through the Horn of Fuca and coming through quite a storm. So everybody got down, and those ones who haven't been at sea, actually had 3 days to recover. from the storm, but it was pretty rough. And however, what you can see as well, there's a high pressure in the middle of the Gulf of Alaska. And that's where we were heading. And for most of the survey, 3 weeks out of 4, I should say, we had this kind of weather, which was very low wind, almost no waves, little bit of waves, and overcast, sometimes sunny, but really, really nice workable weather. To the point that we were able to complete 3 stations per day. And it's including steaming between the stations. And each station from each other were about 16 nautical miles away. So it's about 180 nautical miles steaming plus station work. And what it implies, you'll see more. However, our last week on the grid was a little bit rough again. So the fortune changed and we had a quite rough winds, but we still managed to complete the last about transect and a half. The survey. So 3 weeks, 60 stations, lots of ground cover. You want to add something?
Yeah, this is, so just to say, this is around probably 45, 50 knot winds. So this, so the swell is probably 5, 6 meters. Not very rough, but enough to upset the ship and the crew and the sampling.
Yeah, the good news that this was a kind of overwhelming. That means swell would be kind of cancelled. By the wind would still be strong, change in direction, and kind of swell wouldn't actually go about 6-7 meters. Okay, now you want to talk about this?
Sure. So I was going to say there was a figure there, 700 square kilometers for the survey area.
Yeah.
And that's 700,000 square kilometers.
Oh, 700,000. Sorry, 700,000. Yes.
Okay, so just briefly about the oceanography here, just to say that we're working in the Gulf of Alaska, which is the Alaskan Gyre. So this is it's quite a particular oceanographic region. We have this west to east current coming across the Pacific, and this current splits at around the top of Vancouver Island into the Alaskan current, which goes up our coast, up through Alaska, around the Gulf, and proceeds westwards with some retroflection into this Gulf of Alaska, which creates this gyre. So there's a circular water movement there. And it's subarctic in nature, so it's pretty cold in the wintertime, and it's like down to, you'll see temperature data down to like 5 degrees in the surface, and gets up to maybe 10, 11 degrees in the summertime, depending on where you are. So just to say that there's this larger scale oceanographic process that's going on, and the salmon are actually migrating out and using this dynamic circulation in their migrations. So for instance, sockeye salmon coming out of the Fraser, they move up the coast in a shelf, on the shelf current, and they're swimming with the current and then get into the Alaskan Gyre, and we'll mill around here feeding as we found them, and then head back to the Fraser River. And then it's not just about this larger scale circulation, there's also some more dynamic, smaller scale processes going on. This figure is showing you sea surface height, and the red spots are high spots, and you see these red spots off Haida Gwaii coming off Alaska. So these are eddies that are generated on the continental shelf, and they're moving offshore, transporting water from the continental shelf with nutrients, and particularly iron, which is important for fertilizing this region, which is generally relatively low productivity, and it's iron limited, it's got a lot of nutrients, so the iron being transported by eddies can be important for stimulating production. And we were thinking about this with regard to salmon and how they might be using these high productivity eddies to stimulate growth. And our survey area happened to, so this was February 2019, so this is when we were out at sea pretty much, and our survey area captured these two eddies, and you'll see a productivity signal in the eddies a little bit better.
So the working spaces. It's a fisheries boat, Russian, built in Germany. So sometimes the Russians joke that those tiny little spaces is the payback from Germans to Russians. But it was all workable. So this is just a hydrobiological lab where most of this zooplankton processing was happening. This is kind of another side of the same lab, so this wall and this wall is exactly the same. That's where Brian, myself, and Anton were working, because you can see Anton was analyzing diet of the salmon.
Not everyone can tell that these are salmon stomachs.
This is salmon
But that's
stomachs.
okay.
Yummy.
Yeah.
And we were also working here. This is the filtration lab, which had very few facilities and amenities working. However, that was a hardcore where it was bleached in the beginning, and they did all the filtrations for the HPLC, for the POM, for the chlorophyll, and on that side for genetics, DNA and so on. So everything was done in this room. And our main area of work was actually deck, where the fish preliminary was sorted out, and all other pictures which you will see throughout is actually were done in a geological lab, which was also a little bit bigger than this one altogether. But sometimes it would accommodate 21 scientists, because we've all been on every set, and we all wanted to see what was the catch.
Yeah, so this lab here, so I was working with a guy, Christoph D, who's employed by the Pacific Salmon Foundation. He was doing the real-time stock ID and environmental DNA with me. And so we were allocated the space for the filtration, and as Evgeny said, we had to clean it. I mean, it doesn't explain what a mess this lab was. It was just a storage facility, and there was decades worth of stuff in there that was decomposing. So we spent the first day trying to make it ship-shape and clean for environmental DNA.
This is the best shape you can see.
So yeah, there's a lot of bleach involved. Yeah.
So this is the station routine, and surprisingly enough, it was very well maintained throughout the voyage. That's how we managed to do 60 stations. So average station on the station was about 2.5, maybe maximum 3 hours. But it would compose of about 4 major activities. One is CTD, which would be a rosette going down to about 1,000 meters, and profiling temperature, salinity, chlorophyll, oxygen turbidity, POM, and DNA samples were collected from this. We're going to show you about 6-minute video afterwards how it will work. Then it was 2 Juday nets, that's a special zooplankton net, which Russians use, to about 2 layers to sample, and a Bongo net, which was specifically sampling design was geared towards the Canadian sampling, because that's how Canadians sample, that's how Russians. So we didn't want to have this comparison. However, what was unique about this compared to any other voyage you might have been already, that both CTD and net activities were happening at the same time. So the CTD was going in the front of the ship, and in the middle of the ship you could do bongos and all the nets. And by about 40 minutes later, when we started, all these activities being done. So the rest of the time was actually dedicated to the mid-water trawl. As soon as it's done, we start moving, and we deploy mid-water trawl, which was quite big, about 40 to 30 meters, 30 by 40 meters, and mouth area, which would be towed on average to about 4.5 knots for about 1 hour. And again, I'm going to show you footage, very unique footage, how the fish actually behaves in the trawl. Because these days we all have GoPros. And it would be deployed in the top 30 meters. And while we are retrieving the trout, the Newston net would go into the surface and towed, because we slowed down, and it's about 2.5 knots, and they will be deployed Newston net for 15 minutes off the middle of the ship. And that's it. The station would take altogether from the beginning to the end, usually about 2.5 hours.
For the gear deployments, and then it would take time to process.
Yeah, of course, after this it's all, and then we start moving to another station, and while we're actually trawling 4.5 knots, we're already moving usually towards the next station. So it's like saving time in every single corner we could.
The thing to add is that everything worked amazingly well, despite our maybe some trepidation at the beginning of the voyage, seeing the state of some of the gear. No gear failures, and we missed only 2 deployments the entire survey. It was 2 bongo nets, which we couldn't put in. Evgeny and I decided not to put the net in because the wind was too strong, and we were worried about losing the net.
30 meters per second, so we decided not to kind of risk our bongo net.
So yeah, otherwise everything worked pretty much perfectly. And so just a couple of pictures here, this is, just so you can see what the instrumentation looks like. This is the rosette, Niskin bottles, and CTD over here. Niskin bottles are for collecting the sweet water samples through the water column. And then this is another picture of the bongo net. So this is the Houston net. So Houston net is being towed at the surface. It has floats to keep it up near the surface, and it was brought on board to sample microplastics. And so we did that at every station, and that's the Jude net. The bongo net looks similar to this, but it's just two nets that are Egypt adjacent. And this is the trawl deck, the big winches operating the trawl going out the back down the ramp. And it's hard to get a full impression of a trawl net. The only time it's fully open is when it's in the water. So, but this is the net being brought back on board. It has a long mesh end that comes down to a cod end that collects the catch. And so it's quite a procedure. I think you'll see this in the video. But it's quite a procedure to move the catch, all of the catch down into the cod end. So they bring the net up incrementally, and then you have to lift it in sections. So the catch will fall down, and then they'll pick it up here and lift that up, and the catch will gradually be moved down into the cod end, and then you can sample it.
So I mean, this is some of the catches we had, and how some work. We had some very interesting creatures like frogfish, you know, this right in the middle.
Lumpsucker.
Or whatever, and this is salmon, of course, and this is one of the good sets. Big toffees, a lot of big toffees, mostly during this and squid during the night samples. From time to time we had jellies, and jellies we actually had all the time, but in different forms. And once we even got this black rockfish, rockfish in the middle of ocean,
yeah, so there were some strange things like this big black rockfish, which was in 4,000 meters of water swimming around on the surface. And there was we had one stickleback, which was about 1,000 kilometers offshore, so 1,500 meters.
Yes. Okay.
But just to highlight really that these are the most abundant non-gelatinous animals is metophids and squid, and they were really prevalent in most of the catches. And from a salmon biology perspective, people have gone on a lot about the importance of squid. So the expectation was that we would see a lot of squid in the diets of the fish, particularly coho, Chinook salmon if we caught them, and also sockeye salmon. But we'll show you what the diets actually look like.
Did you get those abundances during the day too? Of squid and coho?
No, they definitely migrated. We don't catch them.
We were only sampling top 30 meters.
Yeah.
So,
but this rockfish was in the smack bang of the
so there's more, and again, we kept some salps, some jellyfish, some kind of what is that?
Lots of fish, Elychosaurus.
And there's different kinds of leptophis. Generally speaking, it was one species, which was really surprising. We would expect it maybe a dozen species, but we only got 3, and one of them was absolutely dominating, like 97%. Okay.
Which has a horrible name, Talatondina crenularis.
Yeah, Talatondina. Sounds like a spell for it.
So this Elychosaurus is an interesting fish. They get really big, and they're mesopelagic, but they feed on. They come up to epipelagic at night. And we caught a number of salmon that had slash marks down the sides. And these fish get up to about 6 feet long. The one we caught them about this size. And they hang vertically in the water.
If you look, this is teeth. It's like Jurassic Park.
And so they hang vertically, and then they slash at prey that's above them. And so when we recovered fish that had huge slash marks and were still swimming around, and we also recovered fish that had scarring, where they recovered from these bites as well. So that was quite interesting.
And something about food. It was Russian traditions, and of course with Russian food. It was really nice to me because it was like going back about 30 or 40 years, my childhood. But some people kind of found it sometimes offensive. Brian can say something about that.
I was surprised that the food was actually pretty good. So I think everyone is I've heard personally a lot of stories about how bad the food is on Russian vessels, and this was a pleasant surprise. And this is our own production here.
Yeah, so this is actually something which wasn't actually in the kitchen, but we caught some fish and we made some mince from this, and we made this dish called puneni. It's like the whole team came together and we did it about 350 of them in about half an hour, I guess. And then we boil them and they look like this, and then you eat them and then drink water.
And Stephen on the wall smiling down at us every day.
Yeah.
Can you make that out? Is that prune? That's prune.
This is the mass way of eating. So this is a scientific table. So this is,
it's a young picture of him. He must have been like in his mid-30s. I don't know why that's
it's
still on the wall.
Photoshop, I guess.
It's definitely Photoshop. Yeah. Okay, so a little bit about the surface circulation. So this is, we're not exactly sure how this circulation...
No, this was from the heads, from the anomalies. That's using model and the sea surface.
Yeah, that's exactly why we're not exactly sure how it was derived, but it's a combination of sea surface heights and models.
Yeah.
But it's demonstrating the strong sort of continental shelf or shelf break currents, and then some eddy features in our survey area.
Yeah.
And then this is actually derived from the CCD data.
This is CCD data, down to 1,000 meters.
Yeah. Okay.
So, and one major feature is that there was a separation here. It looks like that this current going this way, and we observed this bifurcation when that current, subarctic current, is actually bifurcates. The southern part goes to California, and the northern part goes to the Gulf of Alaska. And that was actually then very clear observed in other physical oceanography parameters, like temperature and salinity.
Yes, so this is just the surface temperature and salinity data. So just to show you that there's this gradient across the survey area, north-south and east-west gradients in the temperature and salinity, and it matches the circulation.
Yeah, and the northern part you can see very cold, well, relatively cold, about 5 to 6 degrees. And then southern part is more than 7 degrees Celsius. So, and that division which we put right in the beginning, provisionally, was actually confirmed by this. So this is a oxygen concentration, and only one picture here representing 2.5 milliliters per liter concentration of oxygen. It's the concentration which would impair fast swimming fish. Their metabolism would impair them, their performance. And you can see that the depth of that concentration is different between northern and southern part. It's much shallower in the northern part, about 150 meters, almost to the point where the top mixing layer is, while in the southern part it's much deeper, it's about 300 meters on average.
Yeah, so this is important because it can affect the vertical distribution of animals and the amount of habitat available for high metabolic rates pelagic fish. Yeah, or zooplankton for that matter.
You can say something about that.
Phytoplankton biomass. So this is actually integrated chlorophyll over the top 150 meters, and so I think the main points here are that we had it was very patchy, and that remember those eddies that you could see in the altimetry. This we think was one of the eddies, and there was elevated biomass in that eddy, and this is the edge of another eddy here. And then relatively high biomass in the central part of the survey area. And you'll see how that maps onto the distribution of the fish and the zooplankton and the fish.
But also I'd like to add that when we came out, we did the bottom transept and then we did this transept. And then we started going according to the grid. So when we came here, the concentration was fairly low, but there was no indication that the phytoplankton bloom has started. But when we actually reached this area again, already after about 2 weeks of the good weather, we actually we found that the phytoplankton bloom just been initiated. So we actually observed the time when the phytoplankton bloom in the southern part has started, spring phytoplankton.
Right, so we had someone on board who was doing, their job was to do satellite validation. So they were collecting surface irradiance data, and then they'll use the chlorophyll data that we just showed, but they were collecting some other pigment information that you can use to validate what types of phytoplankton occur, estimating this from satellite products. The types of phytoplankton and the biomass of phytoplankton. One of the problems working in this area in the wintertime is that you have a lot of cloud cover. So it could look like this. All the black is indicating that there's cloud and that you don't have any information. So this is the week of the 18th of February. But as Evgeny pointed out, as you can see from the weather maps, we had this nice high pressure for around 2 weeks of the survey. So we do have some much better coverage, even though it's still essentially winter time in this region. So hopefully, this is just some preliminary downloads that they provided us, but they're going to do a lot of work in analyzing these data and generating final products. Vishnu he's a PhD student of my Sarah Costa. He was working on this. He suffered terribly from seasickness. It was very challenging for him. Okay, this is the zooplankton distribution. So I don't know if you mentioned this earlier, Evgeny, but there was a Russian scientist on board whose job was to process zooplankton samples. And here we just have broad groups, but he was doing species-level analysis of the Judenet samples in real time. And he finished processing the entire survey by the end of our voyage, which was pretty amazing to walk off the ship with all the data in hand. So that enabled us to put some plots together here, just of main groups. Euphorsids are important prey for salmon. Copepods, maybe. Pteropods, as we saw in the diets, were important prey for salmon. And you can see here, what's interesting is that you have these high biomass patches, but they generally don't coincide, these different groups. So there tends to be one dominating in a region over the others. And so we had relatively high euphorsids in the north and in the south. Copepods certainly highest biomass in the south, and the pteropods had a relatively patchy distribution and quite high in the central area, which, if you remember, there was quite high phytoplankton biomass in that area as well.
Overall distribution was pretty, if you combine everything together, it's just very there were no patches, it was almost uniform. So, but if you look into the distribution of particular groups, they're different and they do not coincide. That's, I think, the message out of this graph. Okay, move on. And this is the one. I can take this. This is the picture probably everybody was looking for. So this is the salmon catch of all species in the whole survey. So first of all, we did catch salmon in 83 trawls, in 83% of all the trawls. So that means salmon was caught, and we were anxious when we arrived to the first station, and the first time our trawl was, are we going to catch one, because nobody did it before in this area except Japanese. But Canadians went 1 year or a couple of years, and they didn't catch anything during the summer. And we immediately got salmon, and then it was continuing. Of course, there were some empty catches, and in the beginning we believed we had this pattern that during the night we would catch salmon and during the day we wouldn't. But then this pattern completely broken down, and we started getting salmon during the day and not during the night. So that means those zeros apparently were true zeros. And any amount, if it would be a zero, would be a very good information in this particular case. So, of course, they have an algorithm there, and they, using all the catchability coefficients, they can calculate how much fish we observed in this area, about 700,000 square kilometers. And they counted about 55 million fish of 4 or 5 major species. You can see that about 50% of these numbers belong to chum, about 25% to coho, and about 16% to sakai. And the pink and Chinook were in very low numbers. In fact, Chinook were only caught 3 times, three specimens, three fish. So 55 million, it's a big number. However, all the fish, all the stocks from all the countries. And then we're going to get into the distribution of individual species. The way Russians work, and that's something to adopt in the future voyages, is that they have enough special manpower or specialists on board to process everything. For example, the physics process on board, chemistry is done on board, fish biology is done on board, zooplankton processed on board, and fish diets processed on board. This is the result of the main prey taxa by salmon species, and that doesn't probably include about 50 specimens which we collected in the end, but we do have this information. Anton processed it, and we helped, Brian and myself helped him to ID certain things. What is interesting here is that, well, you see different colors, and this color is representative of FOZs. Like, for example, Sakai and pink would mostly consume FOZs. Could represent a competition between these two species, but hang on to this. Cham is also eating FOZs, but also very diverse diet. Coho was eating something completely different from this, and they were consuming lots of pteropods, while Chinook was feeding on fish and squid. Something we would expect, but there were only 3 specimens, and one stomach was actually empty. And this is how the stomachs look like. You can see that the diet they were consuming was monospecific. It's like only one species here and only one species here. It's nothing else. And this is major prey taxa by salmon species, and it's FOZs. That particular species is Phasa pacifica, which is presented here. And surprisingly enough, that was really a surprise to us, Brian and myself, is that clear fermindata was very numerous and very prominent in the coho stomach contents. The point is that we didn't catch them a lot of those in our net samples. Obviously, we have a completely different approaches to finding food, or something zooplankton. Something zooplankton and finding fish. So they're much more efficient at this. But this is the first surprise maybe came up, and you remember Sakai and pink had very similar diets, but look at their distribution. First of all, there was a very strong separation among species, especially with those species which were feeding on the same thing. So Sakai was feeding here, and pink was feeding here. And Sakai was dominated in the colder part of the survey, while pink was actually in the warmer part of the survey, and very few. Cham was all over the place, and it was probably concentrated highest numbers at the convergence zone, or actually the frontal zone separating cold and warm areas. And coho, it was a surprise to everyone, because everybody believed that coho is a coastal species. Look at this, coho is all over the place, mostly concentrating in the warmer part, but thousands of kilometers away from the coast, wandering everywhere. And when we get a little bit later onto the stock ID of the coho, you'll see that it's actually quite interesting as well. So, and this is the Chinook catches. We had only 3 specimens, 2 of them actually eaten, I guess. Do you want anything to add?
No.
Okay, the second surprise came from the squid. So we had quite a bit of squid, mostly at night, and this is an indication also in the warmer part of the survey. However, it was almost a continuous component in every night toll. However, what was interesting is that 90% of this catch was the Teotis borealis was not eaten by the fish at all. So the idea was that in the past that at least Sakai salmon consumed quite a bit of squid, especially from the data during the summer. However, this time around, first of all, it was not that species which salmon actually prefers, and all this amount of squid which was not actually found in the diet of the salmon. We don't know why yet, but I mean this is quite an interesting surprise to us. Another surprise came from gelatinous part of the catch, of the bycatch of the trawls. Yeah,
I can comment on this.
Yeah. Okay.
Yeah. So yeah, big surprise. Well, I think really not so surprising was salps. We've been seeing salps on the coast for some time, and there were a lot of salps in the southern part of the range. This is Evgeny's pet animal, so you hear a lot about
And
that
actually,
on Friday.
And there are some empty trawls there. We bought in the nets. We can actually fill up all this area. All this area actually shows very high concentrations as well.
There's a definite day-night sampling effects on this distribution as well. So these high points are probably more accurate than nighttime samples. In the northern part of the Sedler area, really, I think that one of the biggest surprises on the entire expedition was this enormous patch of jellyfish, Chrysaora melanasta, that we discovered. Incredibly high densities, and this is the abundance data doesn't tell you the whole story. If you look at the biomass of this, I think what is the figure you had for the salmon? Was it 50,000 tons?
It was about 30-something thousand tons.
So
but overall survey biomass, I think the jellyfish was representing something like 90% of the whole total biomass.
And the estimated biomass was around 1 million tons wet. So it's, the salmon was 5% of that. So it was enormous. And so this is very unusual to find this species, which is more of a shelf species. It's well known from the Bering Sea. It's being recorded currently on the Alaskan shelf. It's very unusual to find it's like this in the open ocean. So it's one of the big stories emerging from this expedition.
Next slide, there will be more of this. So this is the distribution of this a little bit better, just the species. So,
Okay.
So this is the distribution that we found. There was absolutely no overlap between salps and jellyfish, that particular species jellyfish. And we want to comment on the diameter of the bell diameter. So it's a coastal species, and that means the source of this species production should be coming from the coastal environment, because the hydroids should be living there. That means the smaller specimen should be closer to the shore, and the bigger specimen should be closer to shore. But we observed very clear difference. So this is the largest specimens were found here, and this is the smallest specimens were found here. So away from the coast, maybe closer to this one, I don't know. But these specimens were much smaller than these ones, and we have a few hypotheses why it happened. But this was a surprise as well to us.
The expectation is that what we were thinking is that the production is happening in this region here, and there's southward transports as you get to the Aleutian Islands. So you might expect to see the smaller animals here and following the gyre, the bigger animals on the eastern side. But yeah, the opposite pattern doesn't match that idea. So there's
something about the balances.
a bit more to do there.
Balances. So the red dots which are shown on this map is when one of the members of the Russian team was counting salts while we were trawling. And he found out that whatever we catch in a trawl, which is the numbers here, probably underestimated by about 2 orders of magnitude. So the impact what this species might have on the ecosystem can be enormous, and we don't know yet what this impact is. Now there's a few things which we just thrown away because fish people always like to match distribution of the fish with potential like temperature or potential prey. And this is Sakai salmon temperature and FOZ distribution. You can see that there is actually some resemblance between where you see lots of the patches of the FOZs. You can see the high concentrations of salmon. So that's kind of a match. The champ salmon represented here again, it's concentrated in this transitional zone. What is interesting when Japanese scientist Hika-san, who has done voyage in 2006, they stock ID'd the champ salmon. They came up that most of this champ salmon, which is represented here, is about more than 60% of this champ salmon is coming from Asia, Japan, and Russia. So they migrate the whole way through here, and then they return to their spawning grounds. We are actually waiting for the stock ID on that, which is very interesting whether or not it's true. It does kind of match with the squid distribution more slightly. However, it was not touching squid. It did eat some sauce. And the coho salmon. It's actually quite interesting that the really distribution of the coho is coinciding with the squid and with the theropod distribution. But this is a different species of theropod, the one which we didn't catch. We didn't catch much, which was presented in the stories. But still, it's a good coincidence. So, do you want to say something about this? Because
Sure.
It's an interesting piece.
This was one of the pieces that got the most attention because it was the first time it's been implemented at sea is this real-time stock ID. So we had someone who was doing this. It took about 4 days from sampling the fish to getting out the other end information on the stock composition. That's with all the sort of DNA extraction processes, sequencing, and then the bioinformatics to analyze the genetic data and assign it to stock. And yeah, it was really interesting. This is coho salmon. It was interesting to get this information. I mean, everyone's hanging out for this. We're gonna have it for the entire catch in the next month, I think, from the DFO lab. But it was nice to have it at sea. It's the first time it has been done. It was proof of concept of something that can be used more in fisheries management. If you can ID stock in real time, like that day, then you can make decisions on fisheries closures and that type of thing. But for this expedition, it was more of like it was just proof of concept, and it got a lot of publicity for that. But interesting to see that this distribution of fish, basically from everywhere, from British Columbia, Alaska, down to Washington and Oregon. So. It was just this, out there you have this real mix of stocks from all over the Northeast Pacific.
Yeah, and one more comment of this is that there was no special separation by stock. You would expect that stock from Colombia might be prevalent in this area, or Alaska might be prevalent there, but it was all mixed. There was no kind of stock-specific distribution.
So this was only done for two species, coho and Chinook salmon. And the Chinook were interesting. We had these 3 fish, and 2 of them were from Skeena River, which just seemed, no one was expecting to find Chinook out on the high seas anyway. Truth is, we don't catch them very effectively with this net. I think it's pretty easy for a large Chinook to avoid this trawl. But yeah, 2 out of 3 fish were from Skeena River, so they're really indicating this high seas distribution of that particular stock, which was pretty interesting. The other fish was from Snake River, I think in Washington.
Yeah.
Yeah.
Okay. So this is just a few rough, but I mean, in general, I don't want to repeat this because I've already repeated this so many times, but we did this survey and it was possible to do. And it was an international team, and I think all the data shown here is the first like novel data. So that means that voyage fulfilled its need to establish the baseline. So now we have a baseline we can compare all other samplings to. Before, I mean, there was nothing before, but anything after. Okay, and I think the next one is more samples is coming. They do process a lot of samples. The philosophy of the Russian team and Russian way of processing is that they don't bring any sample home. They process everything at sea and they essentially throw everything away. But there was also enormous amount of information collected on fish health, eDNA, stock IDs, and energetics, otoliths, and so on and so forth, which we processed over the lab. And I think that's more or less it. This is kind of better than hand.
It's not true. Some days are like this. Yeah.
So, I mean, he's saying that Professor Kaganovsky, in particular, crew officers and the captain, was actually pretty involved in this. Okay, I think that's all. And now we actually have 6 minutes for show video.
It is day 23, and the Professor Kaganovsky continues to traverse north and south in the Gulf of Alaska. What you are looking at is rare footage. Few cameras, if any, have ever descended into these frigid North Pacific winter waters. and no camera has ever been here before, seeking to unlock the mysteries of the lives of salmon at sea.
It's really amazing work and work together with scientists who really likes what they do.
On this day, the seas are relatively calm, a blessing for the crew of scientists on board who are eager to learn about the fish, their food supply, the threats they face, and the impact of changing temperatures.
I think we're getting incredible and remarkable results.
Does the food supply diminish? Does it increase? Do the changing conditions invite more predators? Already answers to some of these questions are presenting themselves. To date, 55 sets or catches have been made in the southern and northern areas that were predetermined before setting sail.
Рейс своевременный, рейс нужный. Вот. И хотелось бы, что эти работы и наше сотрудничество продолжилось в будущем.
As with all voyages of discovery, some expectations are met and others are not.
Right now I am cutting tiny pieces of fin clips of coho salmon, and I will add these to this plate and incubate them with a buffer, and that will extract the DNA.
So many things that we thought we're finding something different now that we're out here. So new science.
The overarching objective was to determine where salmon go and what happens to them.
I think first word is salmon, since it's a main purpose of our cruise. Success, it's my best hope and my expectation for that.
This is an incredible opportunity for science.
Will they have food sources? Will they be skinny? For chum, the most abundant species caught to date, the answer, as expected, is yes, they are skinny, but they are expected to start eating soon when the spring phytoplankton blooms arrive. Sockeye, on the other hand, are more robust. Many are large and fat and have full stomachs, bright red flesh and eggs, all of which is yet to be understood.
Challenging, exploration, and fascinating.
For the scientists on board, the topic of why there is a different diet among species is a constant point of discussion. Coho continue to mystify the team. They're supposed to be a coastal fish. Why, then, are they appearing more than 1,000 kilometers off the coast, far from their home rivers in BC and Washington state? Then there is the mystery of the pinks. Where are they? This is an odd number year, and they should be in abundance. Instead, they represent less than 10% of the fish caught to date. Pinks have only appeared in the southern reaches of the study area, in warmer water. Central to the survival of all salmon, of course, is their food supply. Zooplankton and phytoplankton are the staples of their diet, and catches of these vital food sources have been relatively large, considering it is still winter. Then add in gelatinous plankton, which has been extremely abundant throughout the survey area, and you have a clearer idea of the nourishment resources the salmon have access to. As well, the team is collecting data to measure the amount of microplastics in the Gulf. What impact and role do they play in the lives of salmon? Understanding what are the driving trends affecting salmon survivability will take months of analyzing the thousands of samples they are collecting. And even then, the voyage of discovery into the secret lives of salmon will have only just begun.
No doubts, it will be probably best cruise what I have my last professional career.
most memorable thing is all science group is very friendly, so I am very comfortable.
Honestly, it's just it's exciting to be out here to do something that has not been done before. So, it's been exciting.
It is my first experience in the large ocean observation.
It's eye-opening to experience this firsthand and to see the fish in action.
So one of the things that came out of putting the camera in the net was we realized, well, as expected, that there's some avoidance. So there were some sets where the camera was there, where we were seeing salmon in the net swimming along like that, and then we brought the net up and there's nothing in the net. So they were avoiding the net.
Actually, that particular image shows about 10 fish coming in and then leaving the net. And then about 3 coming back, and we caught 3. And this all happened in the first 6 minutes of the trawling for 1 hour. So it's just comes as a school. Leaves of the school, it can do that, even at 4.5 knots. And it can withstand the speed of the ship. And the catchability coefficient that the Russians are actually using for calculating how much abundance we get is actually 0.3. So we nearly being spot on for that particular one. And it's very patchy because only for 6 minutes thrown any fish.